So we left off on Tuesday talking about the modes of natural selection. We'd gone through stabilizing, normalizing types of natural selection, and the directional and disruptive. Remember that the disruptive is a rarer model, but it does still occur sometimes. And everything that we're dealing with is genes. Remember, we're talking about these changes that are going on. We're talking about frequency changes in the gene pool. So there are 5 major agents for microevolution. And just think that these things can accumulate to lead to macroevolution, where we start to see new species develop. That's what we're talking about in the chapter 14. We also have an equation to think about, and we'll do some examples, talk about some examples with the equation called the Hardy-Weinberg equilibrium. In science, we don't prove things. We can only disprove things. So the Hardy-Weinberg equilibrium is an equation that if we could have that equation equal 1, then we would be able to disprove that evolution or frequency changes in genes are happening. So let's look at some things that actually disrupt that equilibrium. This is a figure from your book that we're looking at here on this slide, talking about mutations, non-random mating, migrations, genetic drift, and natural selection. This is a nice visual to kind of get you in the right mind frame. You can look at this in your book. But we've got some slides that will deal with each one of these factors independently. So, let's talk about mutations and gene flow. So mutations, if you remember from our lecture on Tuesday, are not necessarily bad things. You shouldn't equate mutations to bad. Yes, sometimes in the population we see the negative effect of mutations, but it's a lot harder to appreciate the many, many positive mutations that do happen. Mutations that might help somebody not get as sick as often. If you don't get the flu every year, you're probably just going to say, well, I'm a really healthy person. You're not likely to say, wow, I have a mutation that keeps me healthy. So mutations are anything that change the genetics, change the DNA bases. And as we go further through the semester, we'll talk about how these specifically happen. There are different types of mutations. There are mutations that just change one letter in your library. Remember, your DNA is your blueprint. I can change one word in the blueprint and mess things up. Sometimes I can have whole pages of the blueprint missing, and that's where we see chromosomal abnormalities. Sometimes I might actually come into that blueprint, something might happen that inserts entire pages of information into that blueprint. And hopefully you can see that sometimes doing that might be a good thing, might give you an advantage, might add a floor to your house. But sometimes if you add in an extra page or take out a page of the blueprint, it can really mess things up. Your final product might not work properly. One thing we see with these mutations is that the rate of them happening is fairly low, or at least having them happen where you can see them, where you can appreciate them. But it is important, and they do have an impact on future mutation or future generations. So we see this genetic shuffling, and we see some individuals who are able to survive some environments better than others. Now, once we get mutations, and once we have individuals who might have something a little bit different, an ability to survive better, or something that doesn't allow them to survive better, then we start seeing those frequencies in the gene pool change. And so the next point that we're dealing with is gene flow, the movement of those genes from one population to another. We also call this term you're probably familiar with migration. An individual goes from one population into a different territory, and then those genes mix with that new population and either assist that population or harm it. So we have regular gene flow that has migrations or things that individuals do, usually with purpose. They didn't just accidentally get blown over there. There was a migration pattern that had to happen. Those are pretty straightforward gene flow issues. What about when it's not by choice? What about if it's not because it's cold and they want to go to a warmer climate? What if it's because something happens? That's where we get genetic drift, the chance alterations of allele frequencies. Something might happen catastrophically. Maybe there's an earthquake, maybe there's a flood, maybe a disease, maybe the plague comes through. It's some kind of catastrophic event that occurs on the population. And usually it's a chance change in allele frequencies. Sometimes some individuals are weaker than the others, and so their allele frequencies get wiped out. But if you're talking about like an earthquake that crushes half the population, there wasn't anything there that would have helped those individuals to survive no matter what. It was a chance, weird thing that happened. If this happens in a really big population, then the effects of this catastrophic event on the gene frequencies probably isn't going to be that major. But if we're talking about a small population, then taking out half genes is a big problem. I have a little picture here that we're going to look at with some penguins on an iceberg. Well, if we have 10,000 penguins on the iceberg and 10% of them die, it's unlikely that any given characteristic, like a red penguin that we see here in the picture, would completely be gone from the population. It might be decreased, but it's still there. So big populations, genetic drift doesn't harm them that much. Look at the B side over here. With only 2 penguins on the iceberg, and only one of them, 10% of the population, carries the red allele. Well, if something happens to half the iceberg and 50% of the population dies, there goes the red allele. No more red penguins. That is gone from the gene pool now. These are the things that happen with genetic drift. Small populations get impacted more seriously by these events that occur. And that leads to bottlenecks. The little penguin picture we saw just previously is a bottleneck effect. We also see this with things like cheetahs in the wild. Cheetahs were hunted to near extinction, and now a bottleneck effect has occurred because of that overhunting. There really wasn't anything that led us to probably kill one cheetah over the other. We just killed them all for their pelts. Now there's very few left. A bottleneck has occurred. Some of the genes that were in their gene pool are gone now. So anything that happens to them disease-wise is very problematic for their species continuing. Another version of genetic drift is the founder effect, where some small subpopulation migrates into a new region. So we have that migration there, a small subpopulation, but then for some reason they don't mix with the new population. They establish this new population and they stay there, and they don't interbreed with the surrounding population. We see this with humans quite a bit, actually. If you think about something like the Amish community, and your book talks about this as well, so go through and read those sections. The Amish community migrated to the United States, but for cultural and religious reasons, they don't tend to interbreed with the rest of the population. They keep to themselves. So they're a small subpopulation. Now, within that small subpopulation, the gene pool is not as diverse, and they tend to only breed with themselves. And so there are some diseases that have popped up within their population because of that lack of diversity in the gene pool. Specifically, the Amish communities have a big problem with SIDS, or sudden infant death syndrome, because of this founder effect. But you can see how that's changing and altering gene frequencies that might lead them to not be as successful in their environment.